Yeah.
Vamsi, you're back in TIFR over
I am.
because I can see the blackboard in the background.
Also, you can see Malay.
Oh, yes.
Vamsi, you can start.
Okay. Yeah. So, good afternoon, everyone. So, in today's uh, talk, today's speaker is Professor Sudipta Maiti. So, he doesn't really need an introduction. But very briefly, Sudipta joined us uh, in year 1998, and he has been with us since then. Just before that, Sudipta was a postdoc from the year 94 to 98 in the lab of Professor Webb at Cornell. Webb was a pioneer of important spectroscopic techniques. Unfortunately, passed away a few days back on October 29th, 2020, at the age of 93. In today's talk, Sudipta will discuss the scientific life and contributions of late Professor Webb in the talk titled Webb Shining Light on Biology. So Sudipta, please, we are very excited to hear this talk.
Thank you very much, Vamsi, and thank you, Jyotishman, for arranging the YouTube webcast. Welcome everybody to this talk from different parts of the world. So this is, of course, about Webb, who I knew as a person, as a teacher for a long time. I've known him for a long time, and sadly, he passed away less than a month ago. So the point of talking about a great scientist such as Watt is not to eulogize him. That, in fact, defeats the purpose. The point is to try to find out what are the things that he did and try to learn from it, because then it is our duty for all of us to try to see what works for us. And that's exactly how I'm going to take it. There will be no eulogies, just looking at his science. Okay. So if there's one thing that describes Watt, unfortunately I have to use the past sentence now, is he was an experimentalist to the core. Okay, and there is in his own words, the thing that he used to say the most and used to be written on in his office. There was quite a few interesting snippets in his office. And this was tentanda via est, okay, which he translated for himself as to experiment is the true way. It's not to say he didn't respect theory, but he tremendously disliked hand-waving model building. As you will see as you go through his work, snippets that I'll talk about, he always looked for the little details in the experiments and their interpretations. And anything just said, oh, it must be working that way because, doesn't that go on or doesn't that reduces or increases, he will absolutely detest. So until something, some theoretical model is proven by an experiment, beyond a doubt, he would keep doing the experiments. So here is an experimentalist to the core. He was also obviously an method builder. He was an innovator par excellence. In fact, to the extent that his innovations are there, I bet, in every research-oriented biology department in the world. There's no research-oriented biology department that I know of that doesn't have a multiphoton microscope. And many chemistry and physics labs in the world, which probably also have, in addition to this or instead of this, has a fluorescence correlation spectrometer. But lesser known are facts that he was the first one, together with his graduate student, Professor Beasley, the first one to stabilize a superconducting magnet and control the current to an extent that it could be changed by one worsted at a time. So there are many inventions that he has pioneered that adorns all of our labs, including, of course, mine. So let me try to describe his science and appreciate his science, and through 3 statements that he used to keep making. First is his passion for impossible measurements. You say something, oh yeah, would it be so nice to have measure that because that will explain this or that, but that's an impossible measurement. And his ears will perk up and he'll say, let me try to see if that's really impossible. He really was somebody who was after measurements, more than, as I will say at the end, more than about the science itself. It's about the challenge of measuring something. And this let him hop between fields, even though he never called himself an interdisciplinary scientist, between fields that are amazing. Anybody, all of us who call ourselves interdisciplinary scientists would be envious of where he hopped from. The second statement we heard him say quite a few times, including in meetings such as the annual Biophysical Society meetings. We did that 20 years ago. That irritated people to no end, I know that. But he didn't mean it exactly the same way. That irritated people. He meant that he evolved a concept, maybe in a different context of science. That concept actually is the same that somebody else is using now. And again, more than the focusing on the science at hand, he was delighted to understand the concept and to translate this concept to some other area. That's what let him hop between fields so efficiently and make a mark in every field. And the third one I already mentioned a little bit. Hey, but it doesn't add up. That's the dreaded statement, most dreaded statement for all of us, the students. We'll describe something and he will think about it, scratch his head a little bit and say it doesn't add up. He will grab a piece of paper, he will put in some numbers, he will put in the solutions of differential equation, let's say for a cylinder, many of those what he knew from his heart, and say, look, this limit is somewhere else. So what he learned from him, and many people did, is to quickly, when somebody describes some science, quickly take it to the limits and figure out if it adds up. And if it doesn't add up, he's not going to have any of that. So an obsession with understanding the details and dislike for handwritten arguments. So this is what I am going to tell you today. First part, I will do a bird's eye exploration of Webb's contributions, of which there are many. He had nearly 400 papers, and I'm going to not be able to talk about more than 4 or 5, not even that. Only 2 I will pick up in some detail. And so those 2 are the most important ones for biology, okay? And I have already mentioned, for chemistry and for biology, one is for measuring chemical kinetics. It's called FCS, or fluorescence correlation spectroscopy. And the other is one of the foremost tools of cell biology today, or tissue biology today, even in vivo biology today, it's multiphoton microscopy. And then with a little bit at the end, depending on the time I have, I'll tell you a little bit about the person. Okay, so let's start with his first paper that he published as a corresponding author. Okay, and throughout all the papers you'll see this theme coming back again and again: impossible measurements. The first one that you see is a nanowire, except those things were not called nanowires in 1957. There's this first corresponding author paper, Webb being Webb, it was in Science, and he was a material scientist at that time. In 1957, he used to work for Union Carbide, okay, the big corporate thing, which later got into controversy due to Bhopal and all. But Union Carbide was also a good research center, and Webb didn't have any intention initially to become a scientist. He graduated from MIT Sloan School with a degree in business and engineering management. His dad was a banker, and he thought he would do something like that. He got a management degree from the Sloan School in MIT, and he went to join Union Carbide, where he joined the research division and quickly rose to become an assistant director for research. And what caught his fancy were phase transitions. Okay, he was involved in underwater arc welding. It's something that Union Carbide was interested in. And he was looking at it with electron microscopes, what is happening, and he found these things, as today I guess we'll call nanowares, these microcrystals, or at those times he called them whiskers, of palladium, the one that you see here, and a few other materials. And he went on to measure the failure stress and showed that the failure stress of these nanowares is much larger. So that they're much tougher than the bulk crystals. Yeah, so
can I ask the question?
Sure. Yeah.
So can you give a bit more details about measuring the stress in TEM? How is it like a combination of TEM and some other technique?
Yeah, obviously some other technique. I mean, EM was also new at the time, 1935. So this is just a picture. I've forgotten how this was. I'm sorry. You can. That's why I have a reference. It's an interesting question. How did he measure the failure stress? I quickly read through the paper. Towards the last paragraph has this failure stresses and says orders of magnitude higher than bulk material. How he exactly did it somewhere in the middle of the paper. It's a short paper. It's a brief. It's a letter kind of paper.
Yeah, it's old.
You'll see that, and probably it's there in some other paper which he refers to here.
Okay.
So it's most likely. That was also another of what's typical things. He has a result. He will succinctly put it up and then then write a very long 30-page article on it so that everybody can see what he's talking about. So that is his first published corresponding author paper. And so the reason that he started doing this, of course, after a while, he realized that he needs to understand some science. He's getting into administrative position in a science measurement unit of Union Carbide. He went back to school in MIT, did a PhD, actually it's a DSc, so did a doctorate of science in MIT on things like this, phase transitions actually, and material science basically. And that's what he came back and rejoined Union Carbide and was doing at this point. So phase transitions is what his the love was to start with. And actually that is one of the abiding love that he had throughout his life. This is the last corresponding author paper from Webb. And this, you'll see, I deliberately picked up a picture where there is some similarity between the two pictures. If you look at it, though there's nothing to do with each other, that's just for fun. It's a curve that shows phase transition in proteins. Protein biophysics is looking at aggregation of proteins. And this is the dissolution model for TIFR incubated green fluorescent protein, GFP. So he shows that there is a phase transition somewhere. And this is 2012 when he retired, and this is his last corresponding author paper. So 55 years, okay? And he was 85 when he did his last corresponding author paper. And he did have this footprint in many areas. So between these two, pure biophysics and pure material science, he obviously touched many other parts. And his next love was when he joined Cornell. Cornell hired him because they started a new material science program in the late 50s. They knew that material science is coming up big. And they hired this bright young guy. And he started working, but there's some other part of Cornell physics which is doing superconductivity. And later on, a Nobel Prize even happened from there. So obviously, he had a nose for anything that's interesting and anything that had a measurement problem associated with it. But this one I'm talking about, his first superconductivity paper, has nothing to do with measurement. It's a pure theory paper looking at experimental data carefully and proposing a model, which he shows by each experiment is the only model that's possible to explain, that can explain all those measurements. So it's a single authored theoretical paper and dislocations in superconductors, and this is in Science also. Sorry. So this is it's in PRL. I'm sorry, this is in PRL, and he at the same time was doing experiments in material science. Of course, now he is no longer yoked to the interests of Union Carbide. He could do whatever he wanted. He looked at ice, and ice phase transition was something that was very interesting at that time. So I show it here because he's thinking about dislocations now, again, material science things. And he has a science paper again showing dislocations in ice. Most of his experiments and experimental paper are very few authors. In fact, most are 2 or 3 authors. He will give a job to one graduate student who will toil for 3 years and then get something good, or sometimes actually not get anything good, which is not the best for the graduate student, but he used to give these deep problems and push for it. He had no, though he published 400 papers or so, he had no this thing, because anybody who has worked with him will tell you, no interest in publishing papers just for the sake of publishing papers. So it had to be something that excited him. So the reason I put those up is it's dislocations that sits in mind, and he is getting into superconductors, talking to his new colleagues in Cornell and looking even at the theory of it. He was very bright, one of the brightest I knew, and he could just put down pen and paper. And as you would have seen in his office picture, the picture that was there in the beginning, his office was full of books, probably 1,000 books or something. And he knew each one of them. He would say, oh, that equation, sorry, I'm forgetting. So out comes this book. He opens almost at the right page and says, here somewhere, here. So that's the kind of guy he was. So then he makes a transition. So material science, superconductors. Then he makes a transition because he's talking about phase transitions, and he goes to condensed matter, soft condensed matter. Okay, so soft condensed matter, here it is something, it's a critical fluid mixture. Because phase transitions, then you go to critical mixtures where two phases coexist, and sometimes you have large fluctuations due to coexistence of the two phases. And he has another PRL here talking about critical fluid mixtures. We have now come to 65, roughly the time he's still, of course, doing superconductivity. He was a very famous person in the field of superconductivity. He left there somewhere in the 80s. Okay, he did it for a long time, and many other people in Cornell did superconductivity, also some of his students there, and many other cases. So he's still doing instability. See, this is the same sort of the basic concept of the thing. As I said, more than the science itself, the basic concept interested him the most. That's what allowed him to hop between fields. We're talking about annihilation instability in hard superconductors. The physics paper 65. The same time, it's talking about instabilities. The critical fluid mixture will show some instabilities in fluids. This is the first fluid paper I'm talking about, so I called it soft condensed matter. Later on, he'll go over to phase transitions in membranes. Okay. So, oops, I'm stuck somewhere. Hold on a minute. Okay. So this critical mixtures, of course, comes with fluctuations. So somewhere in the mid-60s, he started thinking about fluctuations as a phenomenon. As I said, the critical mixtures was with some fluids. He was interested in those fluids, but he was more interested in the underlying the physics of it, which is the fluctuations. So he thought about fluctuations. He knew about superconductors. He knew about filaments because his nanowires he talked about. His intrinsic quantum fluctuations in uniform filamentary superconductors. Okay, it's a two-author paper in PRL, and he's publishing his 1968. And I know in 1968, because he had already started talking in fluids, he has a new colleague, Elliot Elson, who just joined the chemistry department or chemical physics or whatever in Cornell. And he's beginning to think about DNA, but as a fluid, as fluctuations, and he comes up with a measurement technique. Is one of the two things I will talk about, which is in 1972. Again, I know from the horse's mouth that 4 years it took them to get this going. So 1968, when he was publishing these quantum fluctuations, he stopped looking at classical fluctuations and the nature of fluctuation itself in order to measure binding of DNA. Actually, binding of DNA to transcription factors, the problem brought to him by Elliot Elson. The DNA has to open up and close, and nobody knew at that time how the DNA opens and closes. He had no exposure to biophysics before this. This is where his biophysics comes in. He brings in the fluctuations things. And of course, Elson was a gifted physical chemist also. And they come up with a measurement technique which looks at fluctuations and can measure chemical kinetics. I'll come to that. There's the first paper, the invention of fluorescence correlation spectroscopy, PRL, in 1932. 3 authors, the graduate student McDay and 2 colleagues, Elliot Elson and Webb. And it's going to be 50 years in 2 years, and I thought I'll have a big celebrations. At least I'll have Webb on video or something. That's not to be, because this changed the world for many physical chemists. But let me go forward and come back to FCS. So FCS did many things, including diffusion measurements. So you could look at diffusion. So diffusion is now in his mind. Okay, it's again one of his abiding interests throughout the time from late 60s to the time he did science. 2012-13. So this is diffusion, and he wanted to measure diffusion and found out, given the technology that was available to them in the late 60s, early 70s, FCS was extremely painful. It's a very elegant technique, but it was a bit ahead of its time. You didn't have the single photon detectors, didn't have the fast correlators, you hardly had computers. You know, a place like Cornell, you're beginning to have computers. It was a tough game. So he found out, again, it's a measurement problem in the cell or in a solution that he wanted to solve. So he came up with something which was actually invented by Muming Pu in other place, called fluorescence recovery after photo bleaching. But there was no analytical theory for this. You photo bleach someplace, okay, a solution, and if you are doing a point kind of photo bleaching, the photo bleaching recovers because molecules which are not photo bleached come in, molecules which have photo bleached go out just by plain diffusion, and you look at the recovery, the fluorescence recovery of that spot after photo bleaching, and you try to say, okay, this is faster, this is slower, so the diffusion constant must be changing. And as I said, he is not one to have this hand-waving kind of qualitative arguments. So he went deep down, and this is the first analytical paper that people still refer to for analyzing their data in FRAP, fluorescence recovery after photo bleaching, together with Axelrod and others that he did. So again, diffusion measurements. Now he's interested in that. Okay, and he talked about these diffusion things and phase transitions. Now that he is coming into biophysics and soft condensed matter, that brings him to membranes. There's another colleague, Jerry Feigenson, that joined Cornell, and he's a phase transition man in membranes. He's one of the pioneers, and this is how you see that he has this knack of talking to his colleagues, including organic chemists, including quantum physicists, and trying to get problems that are interesting to solve, and somebody is stuck because there is a measurement that cannot be done. So this is phase transition in membranes. He comes in with this completely quantitative photo bleaching technique, and also has FCS, of course, and starts solving phase transitions in biology, in the membranes. Liquid-liquid phase separation, of course, is 2 dimensions. Now it's a big deal now because it's in 3 dimensions. People look at it. Is looking at membranes. What you see here is a masked cells, okay, labeled with antibodies. So this is the first one of the first live cell measurements that Webb did. Of course, live cells biologists are doing. He gets into live cells. Okay, you would think that he is now getting into biology. It's much later that he would leave the physics part of it. So he's doing many things at once. So it was, of course, a big deal because it was not understood that membranes of live cells which are phase transitions are always thought of as a continuum thing. Jerry Feigenson was talking about it, showing it in artificial membranes, because the measurement problem to do it in live cells comes in with FRAP, and this is his first paper in live cells. It's in Nature. Schlesinger, Webb, Elson, and Metzger. Metzger was the person who had the cells and was the biologist in this. So the first quantitative mobility measurement of cell surface receptors. Okay, the absolute first one, and till now, lots of people are doing lots of interesting stuff. For example, Jitu Mayer in NCBS is making many measurements of diffusion of cell membranes, which are yielding data. And still, some of the same things are used, say FRAP, for doing the measurements. And this, of course, opened a huge thing because scientist of his caliber enters live cell measurements, sees a lot of problems, especially coming through the membrane side. And he one by one goes and has a series of papers in the next 5-6 years, PNA, Science, Nature, etc., and shows phase measurements in phase transitions in cells and membranes, membrane dynamics, receptor oligomerization. He knows, he has a nose for this problem. He looks at the problems, says, okay, this is something interesting and we can measure it. So he gets into live cells. But as I said, it's the challenges of measurement that excites him, okay? And all kinds of measurements. He is a guy who is, if you look at the bottom, is still building instruments. There is a versatile superconducting femtovolt amplifier and multimeter in 1971. Okay, he's already doing FCS and all that. Any of us will take one little sub area of FCS and try to do something significant. He's doing FCS, he's doing superconductivity, making femtovolt multimeters, and he comes across this thing which made a big splash in the late 70s. Erwin Nair and Bert Sackmann in Germany measures current through single channels in a light cell. This is how we think. The channels, they open up, they have this step kind of currents, and that's how you go to single molecule biology. Single molecule biology starts from current measurements. But single molecule fluorescence, I would trace back to Webb's FCS, because unless you measure single molecule fluctuations, of course, with poor instruments, unless you get sensitive enough to do single molecule fluctuations, you can't do FCS. But you average over, because you cannot see a single molecule, you average over, that's what gives FCS the power. Later on, of course, other people have looked at one single molecule at a time and got a Nobel Prize for it. But I'll come to that. But there's single channel measurements, and Nair and Sackmann looks at acetylcholine receptors in a live cells and shows that it works, and it seems like a single channel and a single protein. But still there are doubters around. Okay? And Webb quickly picks up that thing. I mean, even now, it's extremely challenging to do single protein molecule patch clamp recording. I'm talking about 1980s, early 80s, right after it was invented. Webb says, okay, maybe we can do that. Look, it's a measurement problem, right? And he knows how to measure femtovolts, etc. So he gets somebody to, one of his students, to build it and show that this is due to single molecules, because he reconstitutes this at a statistical rate that only one molecule could be there in a vesicle. This is in artificial vesicle acetylcholine receptors. This is his data, single molecule patch clamp recording that you see. Each step is one opening up. Sometimes there are two, so you get two opened up, and he settles the issue in a PNAS paper in 1982 that yes, but Sackmann and Nair are looking at a single proteins active in neurons. Okay, so this gets into neurons, and again another measurement problem. So one of his Cornell colleagues comes to him and says, you know, we roughly know how we see. So if we look at the 5 senses, we know how we see, how we do some things, but we can't figure out how we really hear. There are, of course, observations which suggest that you have these hair bundles, and it's, you know, due to the sound frequency, the hair bundles move in a certain way with some amplitude, and that actually translates to a current in the connected neuron. That current is what finally is decoded by the brain, and you hear. And came with a very specific problem. 2 species of crickets are these insects, not the cricket that Virat Kohli plays. 2 species of crickets have very close to each other, the frequencies that they make, the mating calls, close to each other, but one species doesn't respond to that. And it's thought that the cricket brains are not good enough to tell, you know, to sort of internalize and decide. It must be some resonance problem. It just doesn't resonate with their eardrums. Of course, their eardrums, they don't have ears, their eardrums is on their knees, their legs. So they hear through their legs. So it's a problem that you have to do a measurement, which is, and the amplitude that Webb did a back calculation is probably a couple of nanometers of motion. Okay, that's what one single hair bundle will do, and tug on a neuron, and the neuron will open ion channels. So he builds this instrument, okay, again with one of his students, mechanical properties of sensory hair bundles that can do a fluctuation measurement, actual object displacement. He measured to an accuracy of 0.3 nanometers, okay, 300 picometers. So we are talking about 3 angstroms. And this is 1980s, and optically. Okay, they have an interference pattern of a laser light, slightly separated. They look at it, they put it on a quad detector, and any fluctuations of the object that you're talking about, and the 2 light spots are slightly separate by about 200 nanometers there. It is an interference pattern on the quad detector right at the top here, and that quad detector gives you a signal. And he back calculates, does all the noises things, and shows, yes, I have picometer resolution, and he measures, yes, at a certain frequency you have 3.5 nanometer resolution movement. But he is one that is absolutely stickler to things. It didn't prove to be a solution to the cricket problem. That is, both of those had resonance at those frequencies. But that's the tripartite truth. He has solved a measurement problem. And if you go forward, you will see this kind of technique later became very popular in optical coherence tomography, which people are still doing till now. So another measurement problem. And so I'm half an hour up, right? Vamsi, how much time do I have?
Yeah, I think you have
I started late, right?
Yeah, half an hour. Yeah, half an hour.
Okay, good. I'll just try to stop at 5:05. Okay, so then you come to this measurement problems in biology using optics. So the previous one was also from using lasers, but that was just interference patterns, no imaging. Now it comes to imaging. It's another impossible problem. I'll come to it. The two things I have highlighted, the first one was FCS and second one was this. Here, the measurement problem was to go through a sample with light to illuminate a spot that is in the middle of the sample without the light really going through the sample. I know it looks like a tautomeric thing. So what happens if you shine light through a sample and you have fluorescent markers which will give you some signal. Everything that is in the path of the light, no matter how well you focus it, is getting photo bleached. And this is the standard problem. So in the 80s, something else became very popular, which is called confocal microscopy. Brad Amos and White in in Cambridge comes up with a good design. Of course, it is Marvin Minsky's 1953 patent for confocal microscope. Confocal microscope is getting very popular and in the late 80s, but it has this problem of photo bleaching. Go through a thick tissue, everything is photo bleached by the time you looked at one single point. So it comes up with this idea of multiphoton microscope, which does exactly what I said. It really illuminates a point object inside the sample without really illuminating any of the thing in its path. Okay, so I'll talk a little bit about that. And then it starts with 2 photons, showing that a ball of fluorophores is a latex bead, which is fluorescently labeled throughout. You can draw a geometric pattern. This is a 3-dimensional image. You're looking at a cross section, and light comes to this side and goes out to that side. You can draw a geometric pattern with 4 holes and lines joining them, etc., without actually photo bleaching the rest. Okay, so you can photo bleach this, and that, and a little line through here, here, and here, but the rest of it remains fluorescent. So that becomes the invention of multiphoton microscope, 2-photon microscope to start with. It's a science paper by Deng, Strickler, and Webb, 1990. Okay, later on, 2-photon experiment got extended to also 3-photon. I played a little part in it. This is one of my favorite photographs. That's me, for those people who can't recognize me anymore, about 25 years ago. There's Webb. These are my friends, Warren, Becky, and Chris. These 2 are now profs in Cornell, Warren and Chris. Becky is also working in Cornell in some capacity as a researcher. Okay, so I will talk about what Webb has been doing. Of course, later on, and also, as most people nowadays know him as a biophysicist, in fact, as a biologist, because he has tons of papers on brain sections and things which... Yes.
Sorry, after the guilt of slowing the talk, but can I just go back one slide?
Yes.
These 4 spots that you created,
yeah.
How did you do the illumination?
Okay, this is not my paper. This is Deng, Strickler, and Webb. This is done by a colliding pulse mode lock laser. So lasers were not yet in the scene, the ones that you could buy off the shelf.
Yes.
Okay, so there is a colliding pulse mode lock laser, and then they use that. Yeah, things have become much so much easier. So this is so he's making a transition. So he's making a transition from being a material scientist to a superconductor, low temperature physicist, to a soft condensed matter physicist, to a phase transition guy, to a biologist. Or a biophysicist, as you will call them. But look at his in 1988. Is a sort of a look at it. He's becoming a biophysicist pretty much exclusively from the late 80s, early 90s. But this is, I just picked up his 1988 thing. Of course, it's not a clear transition, but look what are the 6 papers he has published. I've colored them differently. All are 1988 papers, okay? So the bottom one, let's come from the bottom, superconductivity. Of course, he's doing thin film thermistors. So he's still making devices for superconductivity measurements between 50 millikelvin and 300 Kelvin. and comes in an Journal of Applied Physics. Still doing material science. 1 over F resistance. Looking at noise, of course, the noise fluctuations are in his mind. 1 over F resistance noise of hydrogen in palladium. It's a physical review paper and PRL paper published in the same year, 1988. Then he is also publishing, another of his graduate students working on soft condensed matter. Okay, measurement of modulated lamellar P beta phases of interacting lipid membranes. Okay, so he's still in lipid membranes. He had a lot of fun with lipid membranes. He had the things I haven't talked about. He has covers of Nature and all that with lipid membrane papers. He's still doing that. There's a PRL. So Journal of Applied Physics, PRL. Then he's talking about technique development, but in biophysics. Okay? Studies on individual cells, cell experiment, but using quantitative digital video fluorescence microscopy. These were new things, some of which he invented, some of which he actually perfected. So it's one of those things, digital video fluorescence microscopy. He didn't invent it, but he got enamored with it, made the best use of that, and he's showing how to use it the best. The rest is cell biology, all with live cells. And he's published a paper in Science, molecular crowding on the cell surface. He's talking about receptors and their crowding, and cytosolic free calcium measurements, Journal of Cellular Physiology, and imaging asynchronous change in intracellular calcium. These are calcium measurements in PNAs. Okay, so same guy working and publishing simultaneously in Journal of Applied Physics, PRL, and Journal of Cellular Physiology the same year. And I know Webb, he'll walk into one. So this is all the basement of the F corridor in the Clark Hall in Cornell. He'll walk into one room, he's doing something else. He walks into the other room and equally excited about that thing, because that student is doing completely something different. So that's it. But from this onwards, he slowly became more and more involved with biology. Of course, he had a huge amount of people from all over the world coming to his lab, because they hear that he has developed this, he has developed multiphoton microscope, etc. Everybody wants a measurement done. So there are a lot of problems, and he gets enamored with the biological problems from about this time. So if I have to put a year on it, though there is nothing accurate possible, it's 1988 or late 80s. Let's call it that. Okay, so let me just take the next 10, 15 minutes in describing and giving you the flavor of the challenges that he faced and the concepts that he evolved using FCS and multiphoton. Okay. So as I said, in each of these, there was an impossible challenge, an impossible problem. In FCS, the impossible problem is measuring kinetics, chemical kinetics, after the reaction is over. Sounds like an impossible problem. You, all of us, measure chemical kinetics. You think about it. You start from a system out of equilibrium. Okay, create a system out of equilibrium. Look at its return to equilibrium. Fit that data. You measure some parameter that tells you either the product forming or the reactant disappearing. Measure, fit some data and say, okay, this is the equation by which it works. And fit a kinetic equation. You have the measurement of kinetics. After it has ended, no further reactions possible. Things have reached equilibrium. You don't measure chemical kinetics, but you can. That's what Webb showed, and with FCS, and I'll talk about that. This comes in the glorious sort of line of chemical kinetic measurement problems. So people, most of you are chemists, you know about Lord George Porter, okay, who had some association with our department also. In 1998, I think he reviewed the department. He got a Nobel Prize with flash photolysis, fast kinetics measurements. Okay, this was microsecond, sub-microsecond flash photolysis. You prepare something at out of equilibrium and look at it. Something I would call more elegant came from Manfred Eigen, who didn't create a real out of equilibrium thing. He imposed small oscillatory perturbations on the system and looked at, so he knew the oscillation frequency and phase of things that he's putting in. It could be light, it could be temperature, it could be something else. And look at some property of the product and look at the phase difference between the two, the oscillatory formation of the oscillatory position of the agent and the oscillatory product formation there. And from the phase difference, talk about the chemical kinetics. This was elegant because you didn't have to create a very far out of equilibrium. If you do something very far out of equilibrium, usually you cannot use that in biology. And FCS went in that glorious tradition one step further. You don't create anything out of it. You measure it. So I'll come to that. But the second thing then after that I'll talk about is the other impossible challenge that I already mentioned a little bit. Shine a laser inside the sample without really going through the sample. Okay, so here is FCS. Let's start with that. So what I'm talking about is chemical kinetics. And you have some sort of free energy diagram here. Some of the molecules are here, some of the molecules are here. And you want to look at the kinetics. You say, okay, put all of the molecules here, and at some point the equilibrium will be established at some rate, and which will have something to do with this potential energy or free energy barrier. And I will be able to, suppose it's Arrhenius, I will be able to tell you what the kinetics is. Okay, it can happen in chemical kinetics where, say, the molecular nature is changing, some bond is being formed or bond is being broken. But equally well, it can happen in physical kinetics. Say some liquid is getting is evaporating, and if you suppose you have water and you cover it with something, and the temperature is, say, room temperature is all equilibrium. It started with no water vapor there, and then it will establish itself a closed system. Suppose boiling water you just poured in and covered it and left it. What will happen is that slowly water vapor will establish a vapor pressure at that temperature in that constitution of air. It has some certain vapor pressure. Once the vapor pressure is established, there will be no change in the net content of water vapor in that space up here. Okay? So the physical transition is the amount of water vapor that was there in the beginning, which was nothing. Let's say you flushed it with air, dry air, and then you finally measure how much is there, and approach to that would be the kinetics of evaporation. Very good. So, but nothing changes. Nothing changes after equilibrium. Here, I showed you an equilibrium thing, 4 particles here, 3 particles here, because that's roughly at room temperature KT. Let's say this is a little lower, this is a little higher. And here you have some vapor and some particle. But if you look at it, actually the thing is not still. Okay? If you look at the vapor, some of the random molecules, as all of you as students who have gone through the undergraduate, some of the water vapor molecules are hitting the surface of the water and getting into the water. Okay? What keeps the equilibrium is that some other molecule somewhere at the surface of the water is going into, is catching enough kinetic energy to escape that and go into the vapor phase. On an average, equal number of molecules go into the water phase and equal number of molecules comes out. But the molecules are dumb things, right? They're not talking to each other saying that, hey, you go in, I'll come out. So it just happens on an average. So if it happens on an average, at any instant, you're a tiny bit out of equilibrium. Okay? So you're supposed to have 100 molecules in this, let's say, for example. You have 1 or 2 molecules going, you have 98. Nobody told these molecules at the precise same time they have to go back up, and they don't. So ultimately they do, because there's a rate here and there's a rate here, but the rate equations are all bulk equations. So at any precise time, if you make an instantaneous measurement, you find the system always out of equilibrium. In fact, that's what statistical mechanics is. We always get to the average, but if you draw the phenomenon, there'll be width about the average. And this width is something we take as the bane of the experimentalist. We do many, many, many measurements because I'm interested in the average. And the things fluctuate and I get a distribution. Finally, I fit the distribution, get the average. But that distribution has tremendous amount of information. In fact, even about the kinetics of this process. So as long as you have any temperature, in any given temperature, things are always fluctuating. What I talked about was population, but there could be other kinds of things. There's random translational motion of these red things which go from here to there. Or if you are talking about a protein, the random rotational motion of the protein, which is measured by rotational anisotropy. Again, the average is measured by rotational anisotropy, or simple fluctuations of protein parts. All the time, every time. You look at the structure of the protein as comes out of a PDB file, for example, of an X-ray crystallography measurement, and you think that's how the protein runs. No. Even if you give it to somebody like Ravi Venkatraman and said, okay, how is the protein looking like? You'll see things are jiggling about all the time. So there is fluctuation about the average all the time. The problem is, can you measure these fluctuations, little fluctuations, and what can they tell you? Well, as I said, this little fluctuation, this relates to something called the fluctuation dissipation theorem, these little fluctuations actually report the same chemical kinetics that the bulk departures from equilibrium, as long as the system remains linear. Okay. So for small enough fluctuations, the system is typically linear. So if you can look at the characteristic time, as I said, if I go back to that example of the vapor and the liquid, fine, you know that average should be 100 water molecules in the vapor phase, and you measure 98. You wait, it becomes 99, it becomes 102, it comes back to 100, it goes to 97. But if you look at the characteristic time it took to go back from, say, 98 to 100 or 100 to 102 and come back, you'll see that characteristic time is given by the kinetic energy, the velocity of the vapor molecules and the component towards the water layer. Same thing happens in diffusion. Okay, if you have you think a uniform solution of fluorophores in a solution, but if you look at a tiny area, things are going in and going out of that. So if you're able to measure these fluctuations, in one case, let's say individual water molecules, or in another case, individual fluorescent molecules, you'll see the fluctuations about some average. So there'll be some average, and if you can measure those fast, tiny fluctuations due to departures of individual molecules from the solution, from that volume, you'll see something is incessantly changing. If this happened in a bulk scale, in a fluorimeter, you will say, throw that fluorimeter, who is that who sold me the fluorimeter, let me get my money back. But this is actually happening at a microscopic scale all the time. Okay? So the question that you ask is that, how long does this pattern remain self-similar? If you have an average here, and it has gone below average, how long does it remain below average? And Webb is somebody who knew all about statistical mechanics and phase transitions and all critical fluctuations. He knew the function that you need to use to get that out of this kind of data. And you take an autocorrelation function. I will not describe it. Autocorrelation function is a self-similarity calculations. But from the self-similarity calculations, you can actually get data that tells you about chemical kinetics, about diffusive kinetics, about rotational motion. And this is an experiment. The same experiment, you get time scales of measurements which span 10 orders of magnitude. Vicky in my lab does it every day. And you can see that there is information at every time scale, different types of information, different kinds of information. But this is not possible until you are very good also in measuring the fluctuations. So the other side of Webb, as an instrument builder par excellence, comes out and he knows how to build this instrument, and Elson, McDay, and Webb builds this instrument, which comes out in this paper, and shows that you can measure diffusion and in 3-dimensional or 2-dimensional system, which tells you about size. You can measure size of molecules to sub-nanometer levels in a solution, of course, the average size, hydrodynamic radius, in room temperature, with light, which is 500 nanometers wavelength. So all kinds of notions are shattered, but the physics is fine. The physics always gave you that information. You only thought it was noise. So there's a concept of fluctuations being important that came in. And yeah.
Just I've always been curious about the resolution of these autocorrelation functions and the instrumental resolution in capturing these autocorrelation functions. How have they evolved over time? Like for example, when Webb was making these measurements, clearly different techniques can improve the resolution beyond what is possible using like a single beam, right? Any comments on that?
Yeah, so there is actually no theoretical limit on that resolution, because you're just measuring diffusion in and out of a volume. And the solutions right in the first paper, characteristic of Webb, all the theoretical solutions are given. So now the question of how quickly can you measure it? So to measure a fluctuation at a time scale, your instrument has to be responsive to that time. Webb's time was milliseconds, sub-milliseconds. Now we can measure it down to picoseconds. We do regularly, sub-nanoseconds, picoseconds. In CW light, there's a whole system in CW. There is not an iota of pulsing anywhere. You can do it with pulse light also in some ways, but you can have perfectly CW light, and you can measure picosecond fluctuations in this. And you can get, for example, lifetime, fluorescent lifetime out of those fluctuations. It can also be passed in terms of the fluctuation problem, fluctuation of population between S0 and S1. So the time scale now and the instruments now have evolved that you can go down to the single picosecond. Of course, the point is the fluctuation to average beyond noise, beyond short noise, we have to wait for hours. And this is the beauty of FCS, and this is also what actually people say, oh, but he didn't look at one single molecule. It's true. You look at these fluctuations, average over the population, and slowly the correlation pattern emerges out of the noise. But you can wait the whole day. I did my initial experiments for about a year. I wasted trying to do tryptophan fluorescence measurements, which finally didn't work, FCS. I waited the whole day for one correlation after this. So yeah, so the point is, yes, if you give time, if you can have, and now the instruments are at least 1,000, if not 10,000 times better than what we have. So you can measure chemical kinetics also, as we have shown later. So he did this FCS measurements and invented FCS in the early 70s. Then he got enamored with, because it was a hard measurement, but it was very noisy. He got into this FRAP, and his diffusion measurements then came with FRAP. He didn't forget about FCS, but other people started doing FCS mostly in Europe, and it had a revival in Europe in Rigler's lab, and again Manfred Eigen, who had already got the Nobel Prize with this imposed fluctuations. He got enamored with FCS. There is a lot of improvements in that. 90s, when I came back, as Webb hired him and said, "Hey, how about doing this?" And he started this website. And these are some of the chemical kinetics. But okay, since I have only 5 more minutes, let me get to the next thing, which actually he is mostly known for: multiphoton microscopy. Because, as I said, there's not a single biology lab in the world, but it's solved, that doesn't have one. And frequently they have multiple. I think our biology lab itself has 3. We used to have 2, now we have 1. And so this little place has 4. And you walk into NCBS, they have tons of them. That is half a dozen of them. It is just so useful getting data from deep brain of live animals. Balaji used to be a student here, as a lab in IISc as a professor now, looking at deep inside the brain tissue, looking at multiphoton, looking at learning, the basis of learning and memory. So what is multiphoton microscopy? As I said, this is a version of fluorescence microscopy where you get data point by point. You scan, focus your data, focus your excitation to your point, see how much fluorescence comes out. That's the concentration of your fluor at that point. Move to the next one, move to the next one, and then cover the whole volume like this. It's a slow technique, but it gives very high resolution. So the problem was that as you're focusing on one part, you're coming through by exciting. So the same kind of fluorophore is throughout the tissue. It could be a brain section, or actually like brain where GFP is expressed of a mouse. So you're bleaching throughout. By the time you reach deep inside and you have done the top parts of the brain, they open the skull and put a transparent window there. You have already photobleached very much of it, because there is no way to tell the light, hey, don't go there, don't get there. I'm only interested in this little point. So with one photon, which is a standard excitation, you'll come in with a laser light, focus it, and everything from the, say you're coming from left to right, becomes fluorescent green. Okay. If you look at this beam, is slowly picking up the green thing. It's a solution of green fluorescence. If you have 2 photon excitation, this is his brainchild. You have to merge spectroscopy with microscopy. That was the key to this invention. To look at the energy diagram of the thing, you say, wait a minute, this one photon is linear in its power. So yes, when it's focused, it is higher intensity than here, but still not, as you go out from the focus, it's only falling as r squared. Okay, because if you take a cross section, that's r squared. That's not good enough. You still bleach everything. Suppose I have a non-linear excitation. Okay, there is a concept that actually was invented by Maria Guilbert-Meyer, 1931 theory. And she is one of the Nobel laureates, the real smartest physicist ever. She showed in her PhD thesis that you can have this 3 or 5 years after invention of quantum mechanics, 1931. She showed there is a nonlinear term that is nonlinear excitation possible. Some people in the 60s showed, after laser was invented, that yes, nonlinear fluorescence. But to take that into microscopy and says, well, if I have nonlinear fluorescence, instead of falling with intensity as proportional to intensity, will fall as I squared. And there, if you look at the profile, you hardly, so you come in with 800 nanometer light and excite 400 nanometer fluorophore. The beam is so spread out that the intensity is too low here to excite anything. It only becomes large enough, the intensity, to excite the fluorophores here. And again, it defocuses. So it again doesn't excite anything. So if you look at the excitation profile, you only are exciting here as if the light at 400. So there is no light actually impinging at 400. But the effective light at 400 is only here. It neither comes through here nor exists. So it's just tackle this impossible problem. I have too little time to tell you about that. So I'll come to this. This is the standard picture showing that if you are scanning a little area, say one plane, there is a solid fluorescent label, and you're interested in the distribution of fluorophores in this plane. If you do one photon excitation, you're coming from the top, going for the bottom. What you see after cross-section is a large dark area. Even if you are interested only in this, come with a 2-photon excitation, you'll see everything is fluorescent here, everything is fluorescent here, because the intensity was too low. So he did solve that problem, and one other problem got solved by itself, which is the scattering. The reason that deep brain information and how learning forms, memory forms, was not there because nobody could put a microscope there. Here, people are making microscopes which have working distance of a few millimeters and putting in multiphoton microscopy because scattering goes as 1 over lambda to the power 4. If your lambda has become 2 lambda because you're doing 2-photon excitation, or for 3-photon is 3 lambda, then you have moved the scattering lower by a factor of 2 to the power 4 or 3 to the power. That's a large gain. That means you can go down and still focus the light at a millimeter, 2 millimeter kind of length, where previously you could only go 30, 40, 50 microns. And that has really revolutionized neurobiology. You know, many neurobiologists right now may not know what Webb's name, but pretty much everybody is using it. The little funny story I'll tell you. He was also, you see, his graduation was in management. So everything we are told that you should write down, and if there is something significant in your notebook, get it signed by somebody else, so that if there is a patent dispute, you can show it. There's a patented microscope, and that's not enough, right? Nobody used it. He called Zeiss, and I was already there. I joined because he was doing all these fun things, and I joined from a laser spectroscopy femtosecond laser plan. And the Zeiss guy just laughed. Said, "You want a biologist to use a femtosecond laser? This is not going to fly. I'm sorry." 2008, we bought our second femtosecond laser. Okay, the first one from TIFR grant because it's too expensive. Luckily, I had a grant before from somewhere else. The first femtosecond laser was something else. 2008, I was asking spectrophysics guy, salesman, how many, what fraction gets sold to a biologist? And he says, majority. Majority of the femtosecond lasers get sold to biologists. And Zeiss had to, so a little company called Biorad patented it. Zeiss had to buy whole of Biorad, or at least the microscopy division of Biorad later, in order to have rights to use build multiple microscopes. So anyway, so I'm coming to the end of it. It'll give me 2 minutes. This is what Webb the man. Okay, so I've told you a lot about his science, and as I said, I should only concentrate on science because if he were here, he would say, "Don't waste your time." Yes. So, a couple of his things also used to hang on his in his office. The first one, I wish I had a picture of that. I don't. Never teach a student how to hold the soldering iron. That confused me to no end, and later on, I became a little more familiar. The first time I walked in for an interview and went to Cornell, I saw that one, and then they are asking the question. Later on, when I joined, I asked him, "So what is it?" So he says, "Well, this is about giving the freedom to the student to make mistakes. You may see he's doing something wrong, let him do it. He will learn it the best, and he can learn something more that he can teach you tomorrow that you didn't think about." So this big notice, sort of, it actually was engraved on a metal plate, used to hang on his in his office as you enter on the right side. Never teach a student how to hold the soldering iron. Then a couple of things that he was he kept repeating, his kind of catchphrases he used to say. What does he want? And he would be, of course, later on, he was very famous. He would walk into a Biophysical Society meeting or something, few meetings. He was not a man who would travel around, not a man who would like holding any office, though he was the director for the department, the chairman of the department for years. I know that he is not somebody who would have enjoyed that. But he used to be kind of more, the younger students who knows about multiphoton, etc. Oh man, how great you are, etc. And he detested that. Lean back, lean forward and say, what does he want? He actually detested that. You want to talk science, talk science. Then don't bother telling him how great he is, because he doesn't like it. Science is for science. So that's something that he tremendously respected him for. Scientists, far smaller in stature than him, are actually so, they expect that. They expect good respect. And the third thing is, there's a man, I joined him when he was 66 years old. I left his lab when he was 17. And as I said, he was not a frequent traveler, so he'll be in Ithaca, and there he'll be in his office. Monday to Saturday, 6 days a week, 8 a.m. to 8 p.m. sharp. Okay? Either in his lab or in his office. As I said, he didn't like meetings, and also rarely he would be gone for meetings. You want him, you walk into his office, it's not 8 o'clock yet, you'll find him. And these are the days, those pre-mobile days. So in those 12 hours, he probably gets done as much as some of us he has done in 36 or 54 hours, because you keep looking at the mobile and all that. No distraction. You'll always see him bent down, trying to calculate something, reading, correcting a manuscript, reading for another manuscript for a review, doing something. Just pure awesome. Fantastic. But about a month in a year, he will take off. He was an avid sailor. Okay, sailboat will sail in the high seas. And his favorite saying was, let's have fair winds. Okay, that is, let's have good winds so that you could sail. That's how, apparently that's why you went to the MIT, because MIT at those days had a championship sailing team. And that's where he met the love of his life, Paige Webb, a fantastic woman. And it's actually Paige's passing in, I think, 2010 or 11 that made him go downhill, Webb go downhill. So anyway, so Webb is no more. He had this habit of, Paige had this habit of handwritten, hand-drawn Christmas cards, and you'll see sailing would be a feature. There's something that's still hanging in my board there that I got. Unfortunately, I didn't save everything. There's Christmas cheer and fair winds in the new year. So there's a new year thing. So fair winds is something that he wants. He wanted always. But things that you talk to him, and you'd have debates with him in science. And this is a man who didn't suffer fool's gladly. But he was the first one to admit if he was at fault. He will grump, and then after a day, he'll come back and smile and say, I think you are right. In this card, I found this little equation, which is part of an equation that goes for fitting FCS. And I remember the debate that we had, not me and him. It's something that you're saying this thing will go with different factors of AI, different factors. He was saying that. And I think it was Jerome Merz, another friend of mine who's now a prof at Boston, who said, no, it will go as a sum. And he realized it will go as a sum. And he puts that in a Christmas card. So that's where for you. Well, so may fair winds be in his sail. So goodbye. Thank you.
Fantastic, Sudipta.
Yeah.
Questions?
Yeah, thank you, Sudipta. Any questions, please go ahead. Or you could raise your hands.
Could you go a couple of slides? Sorry.
Yeah, go ahead, Dave.
Yeah, could you go a couple of slides back? Somewhere here. Yes, here. So could you just explain? So you said that something that Stefan Hell used. So you could you correlate to that? I missed that.
I'll correlate. Sorry, I didn't have time, so I went over this. See, using spectral properties of a molecule to do microscopy and to do actually something that I would call super resolution came from Webb. If you look at the width of the Gaussian, suppose it's really a Lorentzian, but it's approximately the Gaussian, the focus, you square it, the width is shorter. So if you're doing 800 nanometer wavelength microscopy, but you are getting actually something which is square root of 2 sharper than that. So you are no longer limited by the wavelength. Typically, if you have a high numerical aperture lens, the limit is about lambda by 2. But it is lambda by 2 squared. Right. Okay, so that is something that he came up with. Also, and of course, if you look at the Nobel Prize citation for Stefan Hell and the others in 2014, you see Webb's name plastered all over it. So obviously, they talked a lot about him. Finally, decided not to give him. His single molecule detection, where Marder in the same year got the Nobel Prize for, single molecule detection, the first time you see by Pearson's fluctuation, you can see single molecule fluctuation for Webb. And single molecule fluctuation became a big deal. However, of course, he didn't see one single molecule. That's what Marder did. 1989, I think, or 79 or 89. Get what a Nobel Prize. So, in several people's inventions which resulted in the Nobel Prizes, you would see that Webb had created these concepts, which actually are something that is much bigger or very different from the science itself, the measurement concept, the tools. And he himself carried it from one field to another, and other people spawned other fields that other people flourished. And that's something which is the mark of a true great scientist. It's a good scientist, one who can make one good measurement or make one good thing. And he thinks about it, tries, fails, thinks about it intuitively and finds success. Very good scientist. But a great scientist is that looks at the concept, looks at the physical chemistry of what's happening, comes up with the rules, comes up with possibilities that nobody would have thought. Look at multiphoton microscopy, look at FCS, and then makes a measurement tool that no matter how many times you tried and failed, you never get otherwise. If you just so that's what.
Yes, hi Sudipta, very nice talk. I think in addition to the science, it's like very motivating that one can do research on different topics rather than only the one which get bored with the age, I guess. In terms of the science, you said one can get the chemical kinetics information after the reaction is over. So I'm not able to follow that. Are you saying the kinetics of the reaction that was over? Or because after over, right, there will be no kinetics.
Yeah. So this last part is not. So reaction is over in terms of it has reached equilibrium. But how do you look at equilibrium? Equilibrium has some properties. Let's say the concentration of molecule A and molecule B has reached whatever their thermal equilibrium is. The molecule themselves, so think about reversible equilibrium like this, okay? AB. So you go away and let it come up to this where there is no further change of concentration. But this barrier is finite and you are at a finite temperature. So this molecule which is at A, 3:4, suppose it's the delta G ordained ratio of the populations. But this particular molecule occasionally will cross over. So this 4 will occasionally become 5. And this molecule will cross over. This 5 will 4 or occasionally will become 3 from this side. So even in an equilibrium solution, you have these fluctuations going on all the time. This is one of the basis of statistical mechanics. You only measure the bulk. But if you look at individual and instantaneous, they're always fluctuating about that. Okay? This is actually a definition to me of temperature. Okay? It will not happen at T equal to 0, but a finite temperature will keep happening because you'll have some barrier and something will cross. And they don't talk to each other. It's not like this molecule crosses over precisely at the same time this molecule crosses over so that the population is kept the same. At any given instant, you will be out of equilibrium. You are always out of equilibrium. So the fluctuations that Manfred Eigen imposed, those are macroscopic fluctuations, are actually inherently there in the system. We don't have to do anything. It's always true.
So I think this is true only for the reversible reaction where you achieve the equilibrium. If the reactions are irreversible, A completely converted into B, then you may not be able to use this particular
technique. Basically, if this barrier is very, very high, then you have to wait infinitely long time to see one go over. That is, so you observe it if the barrier is accessible.
Okay. Thank you, Sudipta.
And this is great for living things and all, because living things are non-equilibrium systems, but they're not very far from equilibrium. Otherwise, it take too much energy. So proteins are not just fixed in some sort of equilibrium state. They're always doing this, and everything else in sense, and also in many reversible chemicals.
Well, just adding on to that, reversibility has a time scale, so that is important also. Reversibility has to be defined by a time scale.
So same thing what Vivek said, that time scale is defined exactly by this and the temperature. Okay, exactly.
Yeah. Okay.
JD, any questions from YouTube? Or?
Not that I see at this point. Yeah, there are no questions yet, but there were like 23 people watching from YouTube.
Okay.
We can just wait for a second. Maybe you can take some more questions from.
Does students have any questions? First yearites, if anybody is here, you have any questions? No? All right, then, I guess, Hamsi?
Yeah, I think we'll end at this point. Thank you so much, Sudipta. It was a very different talk, and
thanks for giving me the chance and inviting me.
Thank you, Sudipta. Yeah.
Very nice, Shilpa. Thank you.
Bye.
Bye.